Students, now let us study about the lipids. Where do we obtain lipids? Where do we obtain lipid and how lipids are formed? Let us study what are the lipids. The substances formed by specific chemical bond between fatty acids and alcohols are called as lipids. The substances formed by specific chemical bond between fatty acids and alcohol are called lipids. Now here this is you can consider as a irreversible reaction, means the lipids that are formed from the fatty acids and alcohol, and when we consider that the digestion of lipids occurs, it is nothing but again it is conversion of lipids into fatty acids and alcohol. Means the fatty acids and alcohol come together, they formed a specific bonding and lipids are formed, and when lipids undergoes digestion, again fatty acids and alcohol are formed. Now let us study that how this occurs. The fatty acids, when they are absorbed in the body, they form certain plasma membrane or production of plasma membrane occurs, and for this a phospholipids are formed. Means when the fatty acids they are absorbed in the body, they are again provided to each and every cell of the body, and thereafter the production of plasma membrane occurs. Not only it is restricted to the formation of plasma membrane, but there are several uses of these fatty acids. Fatty acids, they are used to form the various types of hormones as progesterone, estrogen, testosterone, then aldosterone. These are the various hormones that are needed and they are produced with the help of fatty acids. Now, the fatty acids, they are also used for the formation of the nerve cells. We know nerve cells. We have studied in detail in 9th standard about the structure and the function of nerve cells. So the nerve cells has a covering, and that covering is used of these fatty acids. So this is all about the fatty acids or the lipids. What happens? We are very much careful about the consumption of lipids, and we are advised by doctors not to get more fats. The fats in our body that should be very limited in amount as it gives 9 kilocalories of energy from 1 gram of lipid. Means it is almost more than double. We get 4 kilocalories from carbohydrates, 4 from proteins, and we get 9 kilocalories from the lipids. So they should be limitedly consumed. And if the thing happens that you consume in excess of lipid, then what happens about this lipid? That excess of lipids, they are stored in our adipose connective tissue. We have studied various types of tissues in last class. At that time, last standard, we have studied the adipose connective tissue, which is there under our skin. It is used as a cushion as well. That is the cushion which is important in the absorbing the shocks. As well, it regulates the temperature of our body. So that adipose tissue that is formed with the lipids. Lipids, if they are consumed in excess amount, they are stored in the form of adipose connective tissue. This is all about lipids. Once again, I'll revise that the substances formed by specific chemical bond between fatty acids and alcohols are called as lipids. If the lipids, they are undergone the digestion, then again fatty acids and alcohols are formed. All these fatty acids are absorbed in the body and they are provided to the each and every cell or tissue, then if the absorption of fatty acids is carried out, then the phospholipids are produced, and those are important in the production of plasma membrane. The fatty acids, apart from that, produce or form the progesterone, estrogen, testosterone, and the aldosterone. These are the various hormones. These are the hormones which are secreted in the body in the puberty. Then the lipids that are, if consumed in excess, they are stored in the adipose connective tissue. Now, in the form of adipose connective tissue, this is all about the lipids. Apart from lipids, we need certain things which are used in the traces, but they are very, very important for the body activities. Can I know that which are those activities? You are asked to think. That is, sometimes we get the ulceration in mouth. If hot substances that are to be consumed, we feel very, very burning in our body. That is ulceration in the mouth. Sometimes there is a difficulty in the night vision. All these disorders, they are corrected, and that traces amount are said to be the vitamins or the vitamins. They play a vital role, very, very important role in our operation of the various body activities, and so they are called as the vitamins. Now, vitamins are the group of heterogeneous compounds which each is essential for proper operation of body processes. Means, though it's a group of heterogeneous compounds, many compounds, many different compounds, they come together and form a group that is called as the vitamin. And these vitamins are essential for the proper operation of the body activities. If we have to run all these body activities in a proper way, then we need the vitamins. And these vitamins are of two types. One are called as the fat soluble and another are called as the water soluble. In all, we have 6 vitamins: vitamin A, vitamin B, C, D, E, and K. These are the 6 different types of vitamins. Again, they are categorized in the water soluble and the fat soluble. A, D, E, and K. These are the fat soluble vitamins, whereas B and C are the water soluble vitamins. B and C are the water soluble, so those are most unstable vitamins. We know vitamin C, that is, if vitamin C or B is consumed in a large amount, then there is no harm, because these B and C vitamins, they are soluble, water soluble, and they are thrown out of the body through urine. So there is no much harm if they are consumed in a larger amount. But about the fats, it is very important to consume all these vitamins in a right proportion. They are important in traces, but they are important. If there is a undertake of this vitamin, there are also problem. And if these vitamins, they are consumed on overdose is also harmful. So there can be both harm if they are undernourished or overnourished. We have studied in the process of glycolysis, NADH2 and FADH2 are formed. For the production of this NADH2 and FADH2, the riboflavin, that is vitamin B2, and the nicotinamide, that is vitamin B5, are necessary. So vitamins, all these vitamins are necessary for the proper operations of the body as well as NADH2 and FADH2. These are formed with the help of the various vitamins, that is the riboflavin and the nicotinamide. This is all about the vitamins. Students, many times we experience dryness in our mouth. We know that oral rehydration solution, that is ORS solution, is given to the persons experiencing loose motion. We also experience that sweat in larger amount when we exercise rigorously. So let us study the role of water as an essential nutrient. We know that including the role of water, we consider the importance of nutrients. Water is an essential nutrient. Now, why do we call so? First, understand that our body consists of 60 to 70% of water. Then, each cell contains about 70% of water by its weight. Then, a body plasma that also contains the 90% of water. So, total in all, a body contains on a larger amount the water, and it is very essential. So we consider the water as an essential nutrient, and we must always consume the water in a larger amount. Especially we consume this water in the summer seasons, but sometimes we have the negligence about the intake of water in a winter or in monsoon. So water is important and it is the basic nutrient. Next important, that is fiber. Fibers are also important in our digestion process. We cannot digest the fiber, but there is an important role of fibers in our digestion. And the fibers we can get from the leafy vegetables, various types of seeds, fruits, cereals. All these are important as an important nutrient, fiber. Also, fiber that helps in digestion of undigested food material. In the process of excretion, the fibers are very important. They provide as a roughage. Roughage is provided, and that's why there is easy elimination of our excretion. So that is the important role of fiber. In all, we have studied all of the nutrients. First carbohydrates, then proteins, then fats, then vitamins, and last, that is water. So all these nutrients, they give us energy as well as for smooth operations of body activities. All these nutrients are essential, and all these nutrients are essential in the particular amount. So it is not that you consume the nutrients as per your wish or will. There is a proper balance. If that balance is maintained, then our health is very good. We will be fit and sound. That's the importance of nutrients. Okay. Students, sometimes we get injured and after some time we get cured. Now healing mechanism occurs. When we pick up the flowers, at that time the injury to the plants also occurs. A new individual is formed from existing one. In all these things, a major phenomenon occurs that is called as the cell division. Cell division is an important and essential life process. Now let us study about the cell division. When the cell division occurs, as I gave you some of the examples. Now let us study in detail about where the cell division occurs? First, the cell division occurs when the new organism is formed from the existing one. Second, the multicellular organism that grows with the cell division. And the third, that is the emaciated body grows. Emaciated means the body which is very weak by any disease or by hunger. When the body becomes very weak, it is called as the emaciated body. So emaciated body grows. In all these three things, the cell division occurs. Now let us study what are the types of the cell division. Previously, we have learned this. Again, as a revision, the types, that is the mitosis and the meiosis. We know that mitosis occurs in the somatic cells and the meiosis occurs in the germ cells. Mitosis that occurs in the somatic cell as well as the stem cells, and the meiosis that occurs in the germ cells. Now, before we learn in detail about that, let us know that the structural organization, we already learned this in the previous year, that a cell has typical structure, nucleus, other organelles, and togetherly this occurs. When a cell has to undergo the cell division, the cell doubles, and so the shape also increases, that is enlargement of the cell occurs. As well, if there is the chromosome which is of 2n, it gets the 4n. So when the cell division occurs, before that the doubling of the cell occurs. Now let us study how the cell division occurs. Suppose, consider that if the chromosome is in pair, it has the condition that is called as 2n condition, and if a single chromosome is considered, at that time n condition of the chromosome is there. Now let us study the mitosis. As we have studied that mitosis that is carried out only in the somatic cells or kaikwad, that means the bodily development. When we grow, the whole body except the genital organs or reproductive organs, that is called as the somatic development. So somatic development is carried out in two steps. One, that is the karyokinesis, and another is cytokinesis. Karyokinesis, that is the nuclear division. Karyos means nuclear division, and the cytokinesis, that is the cytoplasmic division. Again, this karyokinesis, that is divided into 4 steps. Sorry. Prophase, metaphase, anaphase, and telophase. So, karyokinesis, that is divided into 4 phases, that is the prophase, metaphase, anaphase, and the telophase. Now, let us study all these phases in detail with the figures. Thank you. Hello students, we have learned that the somatic development or somatic growth that occurs with the mitosis, somatic growth as well as the growth of stem cells that occurs with the mitosis. And again, there are 2 steps of mitosis. One is called as the karyokinesis and another is called as the cytokinesis. Karyokinesis, karyos means nucleus. Nuclear division that occurs is called as the karyokinesis, and the development or the division that occurs of cytoplasm, that is called as the cytokinesis. Now, let us study in detail. There are about 4 phases of karyokinesis, that is prophase, metaphase, anaphase, and telophase. Prophase is the first step of this, and it is called as the longest phase of karyokinesis. In prophase, the coiling of chromosomes occur. Due to the coiling, the chromosomes become short and also it becomes thick one. The chromatids that are formed, and it happens because of the fiber that is called as the condensin fiber. because the condensation of the chromosomes that occurs. Look at here. Prophase, that is the longest stage, and in this coiling or shortening and thereby thickening of the chromosomes occurs. The sister chromatids are the centrioles that duplicate and that moves towards the opposite pole. The next important and very, very important step, that is the nuclear membrane and the nucleolus, starts disappearing. Again, there are three stages, that the early prophase, mid prophase, and the late prophase. So the nuclear membrane and the nucleolus that starts disappearing in the first stage. Then the next is the metaphase. Metaphase means mid stage, or the stage in which the chromosomes, they are arranged parallel at the middle one, at the equatorial plane, at the center. And this also first stage that started with the disappearing the nuclear membrane, and the disappearing of nuclear membrane actually completed in this stage. So nuclear membrane is completely disappeared in this, and the chromosomes that becomes very, very clearly visible. Then the chromosomes, they arrange themselves, they align themselves parallel to the equatorial plane, and this is carried out with the spindle fibers. These fibers, protein fibers, they are flexible in nature and they are used for getting this arranged in the at the equatorial plane. Then the spindle fibers, they are formed between the centromere and the centriole. This is the mid stage, so it is called as the metaphase. This is the second stage, prophase, then metaphase. The next comes, that is the anaphase. In this anaphase, the centromere splits and thereby sister chromatids are formed. These sister chromatids, they get separated from each other and they form the daughter chromosomes. And these daughter chromosomes, they are pulled apart. They are pulled apart and they appear in the form of set of bananas. It looks like the set of bananas. And all these chromosome sets, these are pulled to the opposite pole of the cell. So this stage is called as the pulling and pushing stage, or movement. Pulling and pushing movement occur in this third stage, that is ana stage. Then comes the telophase. Telophase is exactly reverse phase of the prophase. What happens here? That the decondensation that starts. Here the chromosomes are condensed and they become short in the first stage, where in the telophase decondensation occurs, and so simple thread-like and thin chromosomes chromosomes appears, chromosome appears, and in this they become a little bit invisible. The prominency or the prominent structure of chromosome that starts to disappear. In this, the nuclear membrane that is formed, where the nuclear membrane was disappeared in the first stage, again it restarts to form, and the nuclear membrane is formed. Then the nuclei are, two daughter nuclei are formed in this. As well, the nucleolus that is also appeared in each nucleus of the daughter nucleus. Then next, the spindle fibers that completely disappears. Here the spindle fibers were formed in the metaphase to get it pulled. These fibers, now the role of this spindle fiber is over. So the spindle fibers, they completely disappear. And so this is all about the telophase. Here four phases are there, out of which in the three phases the difference and the different structure of chromosome appears, and in the last one, that is the reverse stage. So first stage, that is the prophase, that is reversed. Once again, we shall concentrate this. This is very simple, and we have to study all these phases in not only the mitosis but in meiosis also. These stages we have to see, and we can see all these stages of karyokinesis under electron microscope very clearly, and very keen observation is needed. We are given in our book also, and thereafter we have to observe the colorful images afterwards. I will show you the colorful images, all these. Now let us revise once again. First, prophase, metaphase, anaphase, and the telophase. In the prophase, what happens? That the coiling occur, condensation occur, and due to the condensation, the chromosomes become short and thick one. Then the chromatids, sister chromatids are formed. Then they move towards the pole, opposite pole. Nuclear membrane and nucleolus starts to disappear, and in the metaphase, the nuclear membrane completely disappears. Then the chromosomes becomes clearly visible. They arrange themselves, they line up themselves at the equatorial plane, and with the spindle fibers that are flexible in nature. These are the protein fibers. Then in the anaphase, the chromosome splits, and thereby sister chromatids, they are separated. Sister chromatid are the separated daughter chromosomes that are pulled apart, and it looks like the bunch of banana. Then each set of these chromosomes that moves towards the opposite pole of the cell. In the telophase, it starts decondensing. Thread-like thin structure are there. The chromosomes become invisible. Nuclear membrane is again formed. Then the two daughter nuclei are produced. Then nucleolus is produced in each daughter nuclei. Then the nucleolus disappears, and the spindle fibers, that is completely disappeared. This is the reverse phase of the prophase. This is all about the karyokinesis. Then the second step, that is the cytokinesis, where the division of cytoplasm occurs. It is the division of cytoplasm occurs. New two daughter cells are formed, and at the time, how it is formed? At the time a notch is produced. That notch that gets deepened, and gradually this cytoplasm is divided along with the two daughter cells. Then in case of this occurs in case of animal cell. In case of plant cell, what happens? Instead of this groove, or instead of this notch, a cell plate that is formed, and that is exactly midline of the plant cell, and thereby the cytokinesis is completed, where the division of cytoplasm occurs. So mitosis, that is again in two steps, as the karyokinesis and cytokinesis. Karyokinesis has 4 steps, and cytokinesis is a small step in which a groove or a notch is formed, and thereby the two daughter cells are formed, and the cytoplasm moves in this. This is all about the first stage, that is mitosis.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into 2 identical daughter nuclei. It is divided into 4 key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
Students, in the previous period we have studied about the mitosis. Mitosis that is used in case of growth of body, then it is used for the restoration of emaciated body, that is weakened body, as well it is in the formation of blood set, and if there are wounds, then the healing mechanism to get the wounds healed, this process is used. Now let us study. We have studied in detail about the phases of mitosis. Today we shall study about the meiosis. And you know, meiosis, that is the type of cell division which is carried out in case of the gamete, or it is for the reproductive growth and the gamete formation now, or spore formation. Now let us study the meiosis in detail. The meiosis that is divided into the first step, that is the meiosis first and the meiosis second. In the meiosis first, the recombination occurs and the crossing over between the homologous chromosomes divide and they form two haploid cells. This is the shorter stage in which the recombination occurs, the crossing over between the homologous chromosome, and they divide to get two haploid cells. This is the first stage that is called as the meiosis first. Then second, it is the meiosis second. The meiosis second, that is just like the mitosis. In this, two haploid cells, they undergo the division and by separation, the recombination of sister chromatids occur and thereby four haploid daughter cells are formed. These four haploid daughter cells that are formed, and now that is the gamete formation or spore formation. This meiosis second phase is used. In this, the four haploid daughter cells are formed from each diploid cell, that is 2N cell. Next stage, that is the crossing over of the homologous chromosomes occur and thereby genetic combination occurs. We know the genetic combination occur means in the daughter cell the variation occur, and so it is very obvious, it is very clear that these four daughter cells, they are not at all identical. They have the variation as there is a recombination of genetic material is occurred or the genetic characters are recombined. So for all these four daughter cells gener- uh, by generated by this way, they are genetically different one from the parent cell. All these four daughter cells, they are different from the parent cell as well as they are different from the each other. So this is all about the meiosis second. Once again, I'll repeat. The meiosis first, that is the recombination and crossing over of homologous chromosomes occur and thereby two haploid cells are formed. Second, that is the meiosis second. It is just like mitosis. Two haploid cells, they undergo the division and by separation, the recombined sister chromatids and four daughter cells are formed. This meiosis is used for the gamete formation and the spore formation. Four daughter cells formed. They are formed from the diploid cell, that is 2N cell. All this is the crossing over is genetic combination, and so due to the genetic combinations, all the four daughter cells formed, they are genetically different from their parent cell as well as from each other. This is all about the meiosis second. To get all these processes clear, we are given an activity. That activity we have to perform when our practicals will start, and in them observe the figures. I am going to provide you certain colorful figures for better understanding. So with this, we finish this lesson, life process. Do you understand it? Okay. Thank you.